Hello student, good morning. I am Dr. Ajay Kumar Sinha, Professor of Chemistry. Today I am going to discuss chemical composition of nucleic acid. Yesterday I have given idea, now I will continue. Complete hydrolysis of DNA or RNA yields a pentose sugar, phosphoric acid and nitrogen containing heterocyclic compounds, that is called bases. Means on hydrolysis it will give three products. First pentose sugar, second phosphoric acid and third nitrogen containing heterocyclic compounds, that is called base. In DNA molecules, the sugar moiety is beta D2 deoxyribose, whereas in RNA molecules it is beta D ribose. One is beta D2 deoxyribose in DNA, and in RNA it is beta D ribose. Okay. Then only difference is that one is in OH group is in DNA, OH group is there, and in deoxyribose O will be not there, H will be there, but in ribose OH will be there, H and OH, but in deoxyribose O will be not there. That's why name is given deoxyribonucleic acids. Okay. Then in DNA H will be there, O will be not there, and in the RNA H and OH will be there. Okay. I will tell you what are the different type of bases. DNA contains 4 bases, as for example adenine, that is its symbol is capital A, guanine, G, capital G, cytosine, symbol is C, and thymine, symbol is T. This I told about the DNA, 4 bases. And in RNA also contains 4 bases. The first 3 bases are same as in DNA, but the fourth one is uracil, U. Then adenine structure, I am telling you, here 2 ring is there. One is 6-member ring and other is 5-member ring. Both are attached to itself. Correct? Carbon-carbon double bond is common in both structure. Then in a 6-member ring, two nitrogen is there in 6-member ring. Means 4 carbon, 2 nitrogen. Between the 2 carbon, 1 nitrogen is there. Again between the 2 carbon, 1 nitrogen is there, and 2 carbon is continuous and it is attached with double bond. And first carbon has, from the first carbon NH2 is attached, NH2. Correct? This I told about the 6-member ring. In 5-member ring, 2 carbon is common with 6-member ring. Then out of 3, 2 members are nitrogen, and between the 2 nitrogen carbon is there. Okay, CH. Then C and N double bond is there. Between C and N double bond is there, and other C and N single bond is there. This is actually called adenine. Now come to the guanine guanine. G, guanine. Here what is the difference between the adenine and guanine? Then here again you will find same type, means 6-membered rings and 5-membered ring. Then here all things are same as adenine. Only difference is that with the first carbon, where NH2 was attached, there oxygen is attached. NH2 is not there. Oxygen is attached with carbon. In adenine, with carbon, NH2 was attached with single bond. But now, in the place of NH2, oxygen is attached with carbon and it has double bond. Correct? This is the only one more difference is there, that I told you that between the 2N, one C is there. Then in adenine, there C was attached with H. But now here that in the place of H, NH2 is there. Means NH2 was there in adenine. Here also NH2 is there, but position has changed. Between the NN, carbon was there. Now NH2 is attached with that carbon. Correct? Other things are same. Only you have to check the tetravalency of carbon. Tetravalency of carbon you have to maintain. Correct? As for example, where NH2 is attached with that carbon and N carbon, double bond is there. Correct? And all things are same. CO only, O, that is the new thing is oxygen is joined with carbon with double bond. This is the guanine. Means nitrogen is as it is. Total number of nitrogen is as it is. Extra is oxygen is attached. Now cytosine. Here again you will find that here, but here is there only one 6-membered ring is there. Only one ring is there, that is a 6-membered. Then here again you will find out of 6, 4 are carbon and 2 are nitrogen. And now between the 2 nitrogen, carbon is there, that is attached with oxygen with double bond, CO. Correct? And again that NH2 is attached with first carbon, top, NH2. Then here it is also just like guanine, but only thing is that now only it is 6-membered. 5-membered ring is not there, only 6-membered ring. Then here again in 6-membered ring, what was in adenine and guanine, same number of nitrogen is here, means 3 nitrogen. Correct? Then with first carbon NH2 is attached, and this after that N is there, then C and N double bond is there. From the N, C is attached with single bond, and that bond is again attached with next N, single bond, and C is attached with oxygen, CO. I told you between N and N, C is there. That C is C bond is attached with oxygen with a double bond. Correct? Very simple reaction. And 2 carbon has a double bond. Total 4 carbons are there. 1 carbon is in between the NN, and rest 3 carbons are in continuous. Correct? Then after the first carbon, left side, 2 carbons are there, that is attached with double bond, and each carbon is attached with HH. Means you have to check the tetravalency of carbon every time. Then this is actually cytosine, only 6-membered ring and 3 nitrogens, total 3 nitrogen is attached. Okay, but in ring only 2 nitrogen is there, 1 NH2 is attached with carbon. Now next base is thymine. okay, or thymine, anything you can tell. Then here again, what you will find again here, this here, but here in this thymine T, you will get only 2 nitrogen. Only 2 nitrogen will be there. It is also 6 member ring. But in the ring, 2 nitrogen is there. NH, NH. And one, and this 2 oxygen is attached. With 2 carbon, 2 oxygen is attached. That is the different. Thymine, in thymine, 2 oxygen is there. And both are attached with carbon with double bond. CO, first carbon is attached with oxygen, double bond. After that, NH. And in the 2 NH, middle carbon is there, that is also attached with oxygen. Correct? First is the carbon, second is nitrogen, third is carbon, fourth is nitrogen. Means carbon is in the middle, that is attached with oxygen with double bond. Means 2 oxygens are there attached with carbon. And now that 2 more carbon is there, 2 more carbon is remaining out of 6 member ring. 4 I told you, 1 carbon, 1 nitrogen, again 1 carbon, 1 nitrogen. 4 is over, 2 is carbon. Then that both carbon are attached to itself with double bond. And that carbon is one side, it is attached with first carbon with single bond, and another carbon is attached with N with single bond. Means fourth atom. Fourth atom is N. First atom is C, second is N, third is C, fourth is N. With this N, next carbon is attached with single bond. Next 2 carbons, and those 2 carbons are attached with double bond. And this 6th carbon, you can tell, 6th atom, that is a new things I am telling you, that is attached with methyl group, CH3. Here no NH2 is there, no NH2. CH3 is attached. Correct? CH3 is present. That is called thymine. This base is called thymine. Here methyl group is attached. This is the different. And two oxygen is there. Difference is that here in this thymine, 2 oxygen is there and 1 methyl group is there. Correct? It is attached with the ring. And last is uracil. Uracil, new one, which is present only in RNA. Okay, in RNA, uracil will be present. 3 is common, in both. And that RNA differ in uracil. Okay, uracil is present in RNA. And what is its structure? Then again, it is a 6 member. Here again you will find that here in this ring 2 nitrogen is there and 2 oxygen is attached. Correct? In the ring 2 nitrogen is there. Out of 6 atoms, 2 are nitrogen, and between the 2 nitrogen carbon is there. That carbon is attached with oxygen with double bond. And first carbon, that is also attached with oxygen with double bond. And left side 2 carbons are remaining. Both carbons are attached with double bond, both having 1 hydrogen, just to maintain the tetravalency. Okay, this is the uracil, U. Actually, these 5 bases I told you in the whole nucleic acid or nucleotide, I will tell you nucleotide, you will find only these 5 bases. Okay, you remember always. Now, I will discuss a structure of nucleic acid. A structure of nucleic acid. A unit formed by the attachment of a base to the first position of sugar is known as nucleoside. Very easy. Already pentose is there. Then at the first carbon, actually pentose means oxygen out of 5 member, 1 member is oxygen and rest 4 are carbon, correct? Then here 4 members, then with 4 carbon CH2OH is attached. Then if you will count hydrogen, if you will count carbon, total carbon, then 1 is attached with CH2OH outside, that is the 5th carbon. But in the ring only 4 carbons are there. Then first carbon, with first carbon base is attached. Base. Okay, what I told, any base may be attached. And after that, only with that carbon, one H is there down. And after that, second carbon HOH is there, one H and one OH down. And third carbon again same way, H upside, OH down. Means second and third same position. H upside, OH down. Third also H upside, OH down. And at fourth carbon, CH2OH is upside and H is down. And with first carbon, base is attached. Don't forget. Okay. Then this I told you, this is called actually nucleoside. Nucleoside. SIDE. NUCLEOSIDE. Side. Nucleoside. One more term is there, that is the tide. TIDE. That also I will tell you. In nucleosides, the sugar carbons are numbered as 1, 2, 3, 4, etc. In order to, I already told you 1, 2, 3, 4, 5. Okay, means in nucleosides, the sugar carbons are numbered as 1, 2, 3, 4, means 1 dash, 2 dash, 3 dash, 4 dash, 5 dash, like that, in order to distinguish these from the bases. Okay, these numbers are sugar carbons are numbered as 1 dash, 2 dash, 3 dash, 4 dash, 5 dash, in order to distinguish these from the bases. Okay, when a nucleoside is linked to phosphoric acid, you remember, very important point, when nucleoside is linked to phosphoric acid at carbon 5 position, means 5 dash position, then what will happen? 5 position of sugar moiety, we get nucleotide. Nucleotide, TIDE, when phosphoric acid is attached at carbon 5 outside CH2OH, that carbon. From that carbon, if phosphoric acid is attached, then it is called nucleotide. Nucleotides are joined together by a phosphodiester. Now, actually, many nucleotides will join to each other. I told about the only one. But many nucleotides are joined together. How? Then by phosphodiester linkage. You remember, question is asked. Just why in alkyl, you have seen there will be ester group, RCOOR. But here, it will be ester of phosphorus. In the place of RP can consider. Okay. That's why it is called nucleotides are joined together by a phosphodiester. But here R will be not there. Phosphoric acid is there. Okay, that will join with other. That will join with means here you can tell 2 phosphoric acid will join. All you can say nucleotide will join with phosphodiester linkage. Okay, that I will tell by the structure. I will tell the formation of dinucleotide. Now I will tell you. First structure I will tell that now you have seen that pentose sugar will be there, means O will be there, and this O will join with 2 carbon 1 and carbon 4, and after that, after the carbon 1, 2 and 3 are there, all things will be as it is. At carbon 1, base is there, and carbon 5 outside CH2 OH was there. From that OH, that H has gone, because that has joined with phosphoric acid. Then as a water molecule, that gone. Then now PO4 you will get. PO4 because H3PO4 was there. Phosphoric acid, you know, H3PO4. Correct? Then here just to consider that CH2OH, then that O, from that OH, O will be remaining, but H will go with the distance, or OH anyway, water molecule will go, and both carbon and oxygen will join. Carbon and oxygen will, carbon and phosphorus will join. But here actually, direct link will be not with phosphorus, it will link with oxygen only. Oxygen only because OH and CH2OH. Then you know what will happen? OH, CH2OH, OH is there, and from the acid side H will go. Then as a water it will go. Then oxygen will be negative, carbon will be positive. Then both will join. Then like that way, here you have seen that in nucleotide phosphoric acid attached and base is already there. Then suppose now 2 nucleotides are there, first and second nucleotide. Then again both will join. Then where it will join? Then now I told you in pentose, OH is there at carbon number 3. Carbon number 3, carbon number 2 and 3, both OH is there. Correct? Both down OH is there. Then you consider only first OH, third carbon OH you think. That again you can say there will be these things. It will join with that OH, and again phosphoric acid was there, then that H, phosphoric acid also OH was there, then that again same way that OH and H, it will form water molecule and it will go, and it will join, means oxygen will join with carbon, because here again OH will go as OH minus. Suppose carbon OH has gone as OH minus, then carbon will come positive, and here oxygen will join. Oxygen from the OH, phosphoric acid OH, H gone, then O will be negative. Then this oxygen will join, negative oxygen will join with a positive carbon. Then now 2 unit just joined to each other. Then this with 2 pentose in the middle, phosphate will come. Correct? Then in the molecule 2 phosphate is there. That's why name has been given phosphodiester. Phosphodiester linkage. You remember, 2 units joined, 2 nucleotides joined with phosphate, with phosphate linkage. That is called phosphodiester linkage. Okay. Then this I told you how dinucleotides are formed. Di means 2 nucleotides joined to each other by linkage called phosphodiester. Okay. Now this I told only for di. If many like that way, many you can say this nucleotide can join to each other, always with the third carbon. What happened with first and second? Same way, third and fourth and fifth and sixth, like that way, long chain can be formed. Okay. then it will form a long chain. A simplified version of nucleic acid chain I can understand by simple way. Just like a diester, the second can join with third can join with fourth can join with fifth can join with sixth. Then you will find that in the middle many, you can say, base, sugar is joined with base is joined with phosphate, and only then many this type of this unit will come. Whole N you can write sugar with sugar base and with again sugar with phosphate, like that way N number may be there. But both side that is actually polymer, just like a polymer, then both end, one end sugar and base will be there, and it will be left for phosphate. Sugar, one end is sugar with sugar base is attached, and phosphate is kept opened, okay, because it can join with other unit. And first end again same way, sugar already know, sugar is joined with base and base is joined with phosphate. Means first unit has joined with second unit through phosphate, that you can write, okay, because phosphate is making link everywhere. And so in a simple way you can write first end, you can write that is a sugar that is joined with base, and that sugar is joined with phosphate, correct. And you can one valency will be open for the next joining. Second end sugar and base will be there, and one end will be open for the next joining. But in the middle you can write complete. Sugar is attached with base upside and sugar is attached with phosphate, that is the whole N, correct. Information regarding the sequence of nucleotides. Now here one thing you have to understand, that base may be different, not the same base, but all may join, because they are joined with the sugar. Then that base may differ, 5 bases are there. Then what are the sequence of the bases? Nucleotide, every nucleotide may have different bases. Then what are their sequence? You remember, that is very, very meaningful. Information regarding the sequence of nucleotides in the chain of nucleic acid is called its primary structure. Remember, information regarding the sequence of nucleotides in the chain of nucleic acid is called its primary structure. Nucleic acids have a secondary structure also. One scientist was there, James Watson and Francis Crick. You remember the name of the scientist, James Watson and Francis Crick. Both scientists has given a double strand helix structure. What will be that structure? Secondary structure, that is actually double strand helix structure. What I discussed in for the protein, secondary structure, same way. Here it is called double strand helix structure. Just like I told you that stairs, you have stairs, and this in stairs 2 railings are there, and you are turning up to the 4th or 5th floor, correct, in a parallel way. But that structure is there. Both, I can understand like that, both railing is attached to each both these railings are attached to itself by some bonds, correct, maybe hydrogen bonding or maybe disulfide linkage, correct. Then they are joined with some bond. Then that's why it is called a double strand helix structure for DNA. Generally it is for DNA. This scientist has given structure for DNA. Okay, deoxyribonucleic acid. Two nucleic acids, here what happens, two nucleic acid chains are wound about each other and held together by a hydrogen bonds between pairs of bases. From where bonding will take place? Where? Then N has lone pair of electron. In one chain, always whatever with the base, N will be there. That N can make bond with H of second chain. And second chain is there, that H way, everywhere H is there, then they can join. And due to that, they will, you can say, hold itself. They will be held themselves by that bond, hydrogen bonds. Okay, the two strands are complementary to each other because the hydrogen bonds are formed between a specific pair of bases. Here again you remember, bonding will be between a specific pair of bases. You remember, the two strands are complementary to each other because the hydrogen bonds are formed between a specific pairs of bases. As for example, adenine forms hydrogen bonds with thymine, whereas cytosine forms hydrogen bonds with guanine. Correct? Again I am telling you, as for example, adenine forms hydrogen bonds with thymine, whereas cytosine forms hydrogen bonds with guanine. In secondary structure of RNA, now I am talking, this was what I discussed for DNA, now for RNA. In secondary structure of RNA, helices are present which are only single stranded, not double. This is the difference between DNA and RNA. In RNA, in secondary structure, helices are present which are only single stranded. Sometimes they fold back on themselves to form a double helix, just like a rope. It is a single, but it will turn, then again it can just bind itself. Okay, only one long ring is there, long ribbon, or you can tell chain, but it will, in turning, when it will returning time, it will fold to the, you can say, themselves only. Then here this sometimes this chain helices are present in RNA secondary structure. Helices are present which are only single stranded. Sometimes they fold back on themselves to form a double helix structure, but only one ring chain is there. Only one chain is there, that is important. They fold back on themselves to form a double helix structure. RNA molecules are of three types. You remember, question is asked. RNA molecules are of three types and they perform different functions. What are that three forms? Three types I am telling you. They are named as a messenger, and in shortcut mRNA. It is called M. M means messenger, message, from that M. mRNA. In bracket you can write M-RNA, messenger RNA. Like that way, another name is ribosomal RNA. Ribosomal, R-I-B-O-S-O-M-L, ribosomal. In shortcut rRNA, you can tell rRNA. Ribosomal RNA is written a small r, RNA, correct? And here in messenger small m, RNA. And third one is transfer RNA, transfer, T, small t. Then in shortcut you can write tRNA. Then there are three types of RNA. First is the mRNA, means messenger RNA. Second rRNA, small r, that is the ribosomal RNA, and third one is tRNA, small tRNA, means transfer RNA. Question is asked. Okay. Now, what is the DNA engineering? Not engineering, fingerprinting. DNA fingerprinting. Question is asked. It is known that every individual has unique fingerprints. Our fingers have, it will not match with anybody. That's why they will take thumb impression, because it will not be matched with anybody. All person have different, I can say, fingerprint. Then it is known that every individual has unique fingerprints. Okay, these occur at the tips of the fingers and have been used for identification for a long time. But these can be altered by surgery. But by surgery that can be changed, then it will be a problem. Then how will you recognize a person? A sequence of bases on DNA is also unique. You remember important point. A sequence of bases on DNA is also unique for a person, and information regarding this is called a DNA fingerprinting. Means person you can recognize permanently, it will not change. That is by DNA fingerprinting, because DNA will be fixed for any person. A sequence of bases on DNA is also unique for a person, and information regarding this is called DNA fingerprinting. Question is asked. It is the same for every cell and cannot be altered by any known treatment. You can't make a change by any treatment. DNA fingerprinting is now used for, I can say, 4 purposes. This DNA fingerprinting is used. First, in forensic laboratories for identification of criminals. Correct? Criminals, how will you identify? Then in forensic laboratories, in forensic, FORENSIC, forensic laboratories, then it is used in forensic laboratories for identification of criminals. Second, to determine paternity of an individual, that who is father, any persons have any child who is a father, that because that will not change with son and his father will be the same. Then to determine paternity of an individual. Third, to identify the dead bodies in any accident for comparing the DNAs of parents or children. If any person died, then how will you, the person is died, then how can you tell whose son is this? Correct? Then that time also this DNA fingerprinting is used. And last is to identify racial groups to rewrite biological evolution. In world many type of peoples are there, racial, like South Africa, India, correct? Many countries are there. They look very different. Then just to identify racial groups to rewrite biological evolution. Correct? Then these 4 places DNA fingerprinting is used. Now next things I want to discuss biological function of nucleic acid. Biological functions of nucleic acid. DNA is the chemical base of heredity and may be regarded as the reserve of genetic information. Here you should understand, base decides actually in DNA, nucleic acid, 4 bases are there. Then in different persons or you can say all bases will be not the same. Correct? With person to person, this bases sequence of bases will change. Suppose 4 bases are there. Then in next person, their son, again only these 4 bases continuously sequence will be same. 4 different bases, whatever was there in a sequence, that will be same for the son or grandson. That will not change. Suppose base 1, base 2, base 3, base 4. 4 bases were there. Then in son or grandson, all bases will be in the same sequence. All different bases will be in the same sequence. If it will not match, then you can separate it. This is not from the same family. Correct? Then sometimes I told you 1, 2, 3, 4. 4 bases are there. Then first and 4 are there. Then 3, 2 are there. Like that way. Correct? This is also arrangement. Different arrangement is possible. First 4, 3, 2. Again you can write first 3, 1, 2, 2, 1. Now first 2, 3. First 2, 3 and then 4. Means whatever possibility is there, all are possible. Then in DNA, that what are the sequence, that is important. Okay, only by that basis, because that will repeat, because it is polynucleotide, it will repeat same sequence. Then by seeing that, you can tell who are their gene will be the same. Genetic means all are belonging the same family. Then this I told you the main technique. DNA is exclusively responsible for maintaining the identity of different species of organisms over millions of years. A DNA molecule is capable of self-duplication during cell division, and identical DNA strands and transfer to the daughter cells, means it can reproduce, but sequence will be the same. Okay, that is called self-duplication. Self-duplication during cell division, and identical DNA strands. Strand will be again identical. DNA strand will be again identical, and are transferred to the daughter cells. Daughter cells means here next generation. Another important function of nucleic acid is the protein synthesis. From the nucleic acid, protein can be synthesized, because that nitrogen is there. You know that protein has a very big role. Then this nucleic acid is joined with this also, proteins also, it has a relation. Then here protein synthesis can be done. Okay, and this is the important function. Another important function of nucleic acid is the protein synthesis in the cell. Actually, the proteins are synthesized by various RNA molecules in the cell. In the cell, you will get protein by the synthesis. That proteins are synthesized by RNA molecules that will convert to protein, okay, in the cell. But the message for the synthesis, for the synthesis of a particular protein is present in DNA. Correct? It will be synthesized by various RNA molecules. Proteins are synthesized by various RNA molecules in the cell, but the message for the synthesis of a particular protein is present in DNA. Correct? And this I have discussed biological function of nucleic acid. Now I am stopping here. This chapter biomolecules finished. Next class I will start next chapter. Thank you.